Our journey begins over 4.5 billion years ago. Picture a young Earth still reeling from its violent formation. The surface, a molten sea of magma, glowed with intense heat. Volcanoes erupted, spewing gases and molten rock into the sky. This was no place for life as we know it. Gradually, over millions of years, the Earth began to cool. The molten rock solidified, forming the first continents. Water vapor, released from the planet's interior, condensed in the cooling atmosphere. Torrential rains lashed the Earth, filling the vast basins that would become our oceans. This early Earth was a harsh and alien world. Yet within this chaotic environment, the stage was being set for the greatest story ever told, the story of life. The cooling oceans, rich in dissolved minerals from the Earth's crust, provided the perfect crucible for life's first stirrings. This was the primordial canvas upon which the masterpiece of life would be painted. A canvas prepared by the relentless forces of geology and chemistry, setting the stage for an extraordinary transformation. The question of how life arose from this primordial soup has captivated scientists for centuries. In the 1950s, a groundbreaking experiment offered a tantalizing clue. Stanley Miller and Harold Urey set out to recreate the conditions of early Earth in their laboratory. They filled a sealed flask with water, methane, ammonia, and hydrogen gases thought to be present in the early atmosphere. They then simulated lightning, a common occurrence on primordial Earth, with electrical sparks. The results were astonishing. After just a few days, the water inside the flask turned a murky brown. Analysis revealed that it contained amino acids, the building blocks of proteins, the workhorses of life. This experiment demonstrated that the basic components of life could arise from simple inorganic molecules under early Earth conditions. The Miller-Urey experiment did not create life itself, but it provided the first experimental evidence that the building blocks of life could form spontaneously from non-living matter, a crucial step in understanding the origins of life. The Miller-Urey experiment suggested that the early oceans were teeming with organic molecules forming a primordial soup. But how did these simple molecules assemble into the complex structures necessary for life? One theory suggests that the shallow pools at the edges of oceans played a crucial role. These pools, subject to cycles of evaporation and refilling, would have concentrated the organic molecules, fostering interactions that led to the formation of larger, more complex molecules. Over millions of years, amino acids linked together, forming peptides, the precursors to proteins. Similarly, nucleotides, the building blocks of RNA and DNA, arose from the primordial soup. These molecules, the cornerstones of life, began to self-assemble into larger, more intricate structures. The primordial soup was a dynamic laboratory of chemical evolution. Driven by the relentless forces of natural selection, molecules that were more stable or had a better ability to replicate began to outcompete their neighbors. The stage was set for the emergence of the first self-replicating molecules, the ancestors of all life on Earth. Section 4. The RNA world, the dawn of life. Before DNA became the dominant blueprint of life, scientists believed that RNA played a central role. RNA, like DNA, can store genetic information, but it is simpler in structure and can also act as a catalyst, speeding up chemical reactions. This led to the RNA world hypothesis, suggesting that early life relied on RNA for both genetic information and catalytic activity. In this world, RNA molecules would have self-replicated, evolving and diversifying within the primordial soup. These early self-replicating RNA molecules may have been encapsulated within simple membranes forming the precursors to cells known as protocells. These protocells provided a stable environment for RNA replication and evolution, further accelerating the pace of chemical evolution. The RNA world hypothesis provides a compelling framework for understanding the early stages of life. It bridges the gap between a world of simple organic molecules and the emergence of more complex DNA-based life. Section 5. From RNA to DNA, the first cells emerge. While RNA was the star of the early show, DNA eventually took center stage. DNA, with its more stable double helix structure, proved to be a more reliable store of genetic information. This transition from an RNA world to a DNA world marked a pivotal step in the evolution of life. The first true cells, known as prokaryotes, emerged during this period. These single-celled organisms lacked a nucleus, their DNA floating freely within the cell. Prokaryotes were simple yet remarkably successful, rapidly diversifying and colonizing the early Earth. These early prokaryotes were also responsible for one of the most significant transformations in Earth's history, the rise of photosynthesis. This process, which allowed organisms to harness sunlight to create energy, would forever change the destiny of our planet. The emergence of DNA and the first prokaryotic cells marked a turning point in the story of life. It paved the way for the evolution of more complex life forms and set in motion the chain of events that would ultimately lead to the world we know today.